Hello, everyone, and welcome to lecture number one in our medical biochemistry series on the chemistry of carbohydrates. I am Dr. Muzamil Liaqat Ali, assistant professor of biochemistry. Today, we lay the essential foundation for understanding one of the most abundant and biologically critical classes of biomolecules. Carbohydrates are not merely sugars, they are central to energy metabolism, cell recognition, and disease processes such as diabetes and congenital disorders of glycosylation. As future clinicians and researchers, mastering their chemistry is non negotiable. So let's begin with the carbohydrate chemistry. Carbohydrates are defined as aldehyde or ketone compounds that possesses multiple hydroxyl groups. They are also called as saccharides. This definition is precise. The carbonyl group, the aldehyde at C1 in aldose and ketone usually at C2 in ketoses, combined with several alcoholic hydroxyls, gives these molecules their unique chemical reactivity and biological roles. In human physiology and chemistry, they constitute the primary fuel for the brain, the red blood cells, and exercising muscles, the most abundant biomolecules on Earth. They form the structural backbone of plants, the cellulose, and serve as energy stores in animals as glycogen. The term carbohydrate originates from the French hydrate bicarbonate because they the early chemists observed an empirical formula approximating CH2ON for a pentose N is 5 yields and is 5 yields C5H10O5 for a hexose that is C6H12O6. However, not every carbohydrate obeys this formula exactly. Deoxy sugars, for example, 2-deoxyribose in DNA and amino sugars, or like the glucosamine and glycosamine glycans, deviate this formula. Yet they remain classified as carbohydrates because they retain the carbonyl and hydroxyl features. Nature produces roughly 10 billion tons of cellulose annually. This underscores their quantitative dominance in the biosphere and their centrally in human nutritional medicine. So let's talk about the functions of carbohydrates. Carbohydrates perform their major 5 functions, that every medical student must internalize. The very first function is the primary energy source. Glucose oxidation yields ATP via glycolysis, TCA, and oxidative phosphorylase. The second one is the metabolic intermediates. They supply carbohydrate, they supply carbon skeleton for fatty acids and amino acid biosynthesis. Number three, conjugates. They form glycoproteins, glycolipids, and glycosides. Number four, structural roles. Cellulose and peptidoglycan provide rigidity in plants and bacteria. And number five, that is informational roles. They mediate cell to cell recognition, blood group antigen, immune modulation, and growth factor activation. In clinical biochemistry, disruption in these functions manifests as hyperglycemia, glycogen storage disease, or mucopolysaccharidosis. Now, let's discuss the classification of carbohydrates. We classify carbohydrates by the number of monosaccharides units obtained upon hydrolysis. The very first one is the monosaccharides. They cannot be hydrolyzed further into its simpler structures. Examples are the glucose and fructose. The next is the disaccharides. 2 monosaccharide units linked together by a glycosidic bond. The examples are maltose, sucrose, and the lactose. The next is the oligosaccharides. They contain 3 to 10 units of the simplest sugars, right? The examples are maltodextrin, or the blood group oligosaccharides. Next is the polysaccharides. Polysaccharides contain more than 10 units per molecule, right? Linear or branch in its type, starch, glycogen, cellulose, and dextrine are examples. This classification is clinically relevant because digestive enzymes are specific for each class. Deficiency of lactase produces lactose intolerance, while amylase acts on starch. So, let's talk about the monosaccharides. Monosaccharides are further subdivided by carbon number and carbonyl type. The table displays as trioses that contain three carbon structure, right? That contain three carbon structure, right? Aldoses, the example is the glycerols, right? And the ketoses, that the example is dihydroxyacetone. In tetroses, the example is erythrose, and the ketoses contain erythrolose. And in pentoses, that contain five carbon structures. Aldose sugar is ribose, while the ketose sugar is the ribulose. While the carbon 6 containing structure, that is the aldose sugar is the glucose, and the fructose is an example of ketoses. While the 7 carbon structure haptoses that contain aldose sugar, the glucohaptose, or the ketose sugar, that is pseudohaptose, are the example. In medicine, the ribose is essential for nucleotide synthesis. Glucose is a universal fuel, and the pseudohaptose 7-phosphate appears in pentose phosphate pathway. Aldoses. Aldoses contain terminal aldehyde, CHO group, right? Ketoses contain a keto group, that is CO group, almost always at C2 or carbon number 2. Glucose is an aldohexose. Fructose is a ketohexose. This distinction dictates their activity. Reactivity, aldoses are stronger reducing agents and precipitate or participate in Maillard reaction relevant to diabetic complications, right? Let's talk about the hemiaxial and hemiketal formation, right? Open chain sugars are unstable in aqueous solutions. An aldehyde reacts intramolecularly within a hydroxyl group to form a cyclic hemiacetal. A ketone forms a hemiketal. For glucose, this carbon number 1 aldehyde typically reacts with the C5 hydroxyl to create a 6-membered pyranose ring. Fructose forms a 5-membered furanose ring. This cyclization generates a new chiral center at the anomeric carbon, that is C1 in aldose sugar and C2 in ketoses, right? Producing alpha and beta anomers. These ring forms predominate more than 99% in solution and are the biologically active species recognized by enzymes and transporters. Let's talk about the isomers in sugars. Sugars show 4 major types of isomers. The very first one is the stereoisomer. Same formula, molecule formula, different spatial or arrangement. Right? The next one is the epimers, different in that or different at a single chiral carbon, except the C1. Right? The next one is the enantiomers, mirror images like the D versus L. The next one is the optical isomers, rotates plane polarized light. So we will examine each in detail because stereospecificity underpins enzyme action and receptor binding in human physiology. So let's talk about the sugar that shows various forms of isomers. Glucose. Glucose possesses 4 asymmetric carbons, C2 to C5, right? Theoretically yielding 2 into 4, that is 16 stereoisomers. So 8 is D series and 8 L series, right? In Fisher's projections, the carbonyl is placed at the top. The configuration at the penultimate carbon, that is C5 in hexoses, determines D or L designation by comparison with the D or L glyceraldehyde. So D or L system is a configurational nomenclature, not an optical rotation label. If the OH on the highest number asymmetric carbon lies on the right in the Fisher projection, the sugar belongs to the D series. Left yields the L series. Virtually all mammalian monosaccharides are D isomers. Enzymes are stereospecific hexokinase, phosphorylase, only D-glucose, not L-glucose. This is why L sugars are largely non-metabolized and used as low-calorie sweeteners. For molecules with the multiple chiral centers, the D or L prefix refers exclusively to the asymmetric carbon farthest from the carbonyl. Most naturally occurring sugars are D. In clinical laboratories, the naturally occurring glucose is dextrorotatory, the positively signed, right? Hence, the synonym is dextrose, right? Let's talk about the optical activity. Plain polarized light passing through a chiral sugar solution rotates either clockwise, dextrorotatory, or counterclockwise, liberorotatory. Rotation direction is independent of D or L designation. Naturally, occurring fructose is D, fructose, yet it is still metabolized via fructokinase. Optical activity is exploited in polarimetry to quantify sugar rotate concentration in biological fluids. Let's talk about the structure of a simple aldose and a simple ketose. Examine the open chain Fisher projection, right? Glyceraldehyde L-triose versus dihydroxyacetone, or that is ketotriose. The structural difference at the carbonyl dictates distinct metabolic fates. That dihydroacetone phosphate enters glycolysis directly. The next is that D and L sugars are mirror images. So D glucose and L glucose are enantiomers, non-superimposable mirror images with the identical physical properties, except optical rotation and biological activity. Enzymes involved to recognize only the D or L. D form L glucose is excreted unchanged. This enantiomer selectivity is a cornerstone of biochemical specificity. So, next, Epimers. Epimers differs in configuration at exactly one chiral carbon, other than the anomeric carbon. The most medically relevant epimers of D-glucose are the D-mannose, the C2 epimer, that is a component of a glycoprotein. The D-galactose, the C4 epimer, a part of lactose and glycolipids. Its accumulation in galactosemia causes cataract and liver damage. The epimerases interconvert these sugars in vivo. For example, the UDP glucose 4 epiisomerase. This diagram contrasts enantiomers. The mirror image D and L glucose with epimers, glucose versus mannose at C2, glucose versus lactose at the C4 level. Note that epimers are diastereomers, not mirror images, and therefore possesses different physical and chemical properties. Now, let's talk about the aldose ketose isomerism. Glucose, aldohexose, and fructose, the ketohexose, share the formula or the molecular formula that is C6H12O6, but differs in carbonyl position. In alkaline solutions, they interconvert via the Lowry D prion when enkestin transformation through a common anidiol. This isomerism explains why both sugars reduce alkaline copper reagent, Benedict test, and why fructose is metabolized via fructokinase, bypassing phosphofructokinase regulation in the liver, an important consideration in fructose-induced metabolic syndrome. So we have completed our systemic journey through carbohydrate nomenclature, classification, structure, and isomerism. These concepts are the bedrock for the subsequent lectures on glycolysis, glycogen metabolism, and pentose phosphate pathway and the glycoprotein synthesis. In the next lecture, we shall explore the ring structure, meter rotation, glycosidic linkages in depth. We'll review the Fisher projections and DRL assignments daily. So they appear repeatedly in clinical cases, right? So questions are welcome in the comments. So thank you for your attention, and I look forward to seeing you in the lecture 2. So stay curious, stay precise. If you like this video, please like, share, and subscribe to the channel for more videos. Thank you very much.